Good day, class. We'll be talking about carbohydrate metabolism. That is BCH 311. And I'll be guided with the following outline. We'll talk about digestion of carbohydrate, absorption of monosaccharides, entry of glucose into cells, metabolism of sugars by colonic bacteria. We also talk about dietary fibers, and also then we'll now go into the metabolism proper, which is glycolysis as a part of it today, and talk about its regulation. Digestive mechanism for carbohydrate. Carbohydrate digestion starts in the oral cavity of the mouth by the action of salivary alpha amylase. Alpha amylase is an endoamylase that is composed of a single polypeptide chain. It is stabilized by calcium ions and activated by chloride ion, and it has an optimum pH of 7.1. And the enzyme, being an endoamylase, acts on alpha 1,4 glycosidic bonds or glycoside bonds, endo-alpha 1,4 glycoside bonds of dietary starch and glycogen. So what that means is that it cleaves the glycosidic bond between carbon 1 and carbon 4 within the carbohydrate chain, within the starch chain or carbohydrate polymers. That is where it cleaves. The enzyme is incapable of hydrolyzing alpha 1,6 glycosidic bonds or glycoside bonds of polysaccharides. Now, by the action of this salivary amylase, the polysaccharides are split into limidestrin, maltose, and small amount of glucose. Dietary disaccharides, the major ones of which consist of sucrose, lactose, they are not cleaved in the mouth, In the stomach, alpha amylase becomes inactivated by the gastric component of that is the gastric secretion, which consists of hydrochloric acid majorly, inhibit the activity of salivary alpha amylase. So the digestion of carbohydrate stops or ceases. Now, the hydrolysis of polysaccharides, that including the alpha limidestrin formed in the oral cavity, and of the disaccharides, monosaccharides, proceeds to the intestine. They are moved to the intestine. The enzymic activity is favored by the neutralization of the acidity or of the acidic food by the hydrocarbonate, that is the bicarbonate. The bicarbonate are a component of secretion from the pancreatic juice. So the component of the pancreatic juice, which consists of a bicarbonate, the bicarbonate is an alkaline medium that now helps to neutralize the acidity of the food content coming out of the stomach into the small intestine. So that makes up the pancreatic juice together with the bile secretion as well. Now, the intestinal hydrolysis of carbohydrate is carried out by the enzymes of the pancreas and of the intestine, because the pancreas secretes enzymes like what? Pancreatic alpha amylase, and other host of enzymes are also secreted to do the hydrolytic or to complete the digestion process. The former enzymes are pancreatic alpha amylase, like I've mentioned before, and oligo-1,6-glucosidase. Now, within a short span of time, usually within 4 to 5 minutes, pancreatic alpha amylase hydrolyzes the delivered starch and glycogen to alpha limit dextrin, maltose. Hydrolysis of the alpha limit dextrin is effected through the aid, that is with the help of oligo-1,6-glucosidase, which is capable of specifically breaking the alpha 1,6 glycosidic bonds. The bonds at the glycosidic bond or the glycoside bonds at the branch point of polysaccharide. Maltose is formed as the end product of this hydrolysis. Now, disaccharide undergoes hydrolysis in the intestinal wall rather than in the intestinal cavity, and the monosaccharide forms are immediately absorbed. They are immediately absorbed. So these are some of the enzymes that are involved in the hydrolysis of disaccharides. You have maltase, that's for maltose; sucrase for sucrose; lactase for lactose, and their end products are glucose, fructose, and galactose, respectively. Now, the end product of this monosaccharides digestion, of carbohydrate digestion, which are usually monosaccharides, are usually absorbed. Here, figure 1 here shows the overview of the digestion of carbohydrates. As I've explained before, we have the salivary amylase working on starch here in the mouth and all of that. And the limit dextrin, isomaltose, and some other disaccharides are absorbed, move directly from the mouth into the stomach, and like I've mentioned before, the disaccharide do not undergo any hydrolysis because the enzyme are not active in the mouth, so directly they are moved to the stomach. Then, as I've said before, the digestion process is inhibited because of the acidic content of the stomach, until the pancreatic juices secrete the bicarbonate together with alpha amylase that neutralizes the acidic content that are moved from the stomach into the small intestine, where the digestion process resume again. Now we talk about the absorption of monosaccharide. Now, once the carbohydrates have been split into monosaccharide, they are transported across the intestinal epithelial cells, usually via facilitated glucose transport and secondary active transport, and then from there into the bloodstream for distribution to all tissues. Now, the glucose molecules or the other monosaccharides inclusive are not moved directly into the small intestine or into the bloodstream. They don't undergo what we call simple diffusion. No, they undergo a process of facilitated diffusion that are aided by transport proteins, glucose transport protein, which falls into two categories: those that depends on energy, or those that depends on the concentration on the energy gradient created by sodium ion transport, and those that do not depend on that. Now, not all the complex carbohydrates are digested at the same rate within the small intestine. Some carbohydrate sources leads to a near immediate rise in blood glucose level after ingestion, whereas others slowly raise the blood glucose level over an extended period after digestion. Now, the glycemic index of a food is an indication of how rapidly blood glucose level rises after consumption. Glucose and maltose have the highest glycemic index of 142, with white bread defined as an index of 100. The absorption of sugar mostly takes place in the duodenum and the upper jejunum of the small intestine. The relative rate of absorption is shown here. Of importance, you see the rate of absorption. Glucose is absorbed 100, galactose 110, fructose 43, mannose 20, xylose 15, and so on. And this indicates that galactose has the highest rate of absorption, followed by glucose, and so on. Now we'll talk about this. Figure 2 shows the transport mechanism of glucose. From the small intestine. Now, how it is moved from the small intestine into the cell, into the intestinal mucosa cell, and from the intestinal mucosa cell into the blood streams, into the blood. Now, from the intestinal mucosa cells from the intestinal lumen into the intestinal mucosa cell, it is by secondary active transport, meaning it involves the use of energy gradient that is created by the sodium potassium ATPase, or what we also call the sodium potassium pump, that the energy gradient created in moving 3 molecules of sodium ions out of the intestinal mucosa cells into the blood and then moving 2 potassium ions from the extracellular space into the intestinal mucosa cell. The energy gradient created is then used to couple the transport of glucose along that concentration gradient as sodium ions flows back into the intestinal mucosa cell down its concentration gradient. So that energy gradient created by the sodium potassium ATPase is used to move the glucose into the intestinal mucosa cells. Now, from the intestinal mucosa cells into the blood capillaries, is by facilitated glucose transport using that is sodium independent. So but this other one depends on sodium. Now we talk about the entry. We are still on the entry of glucose, as I've said, with the explanation of the figure 2 there. We have the ones that are insulin dependent, as I've said before, and the ones that are insulin independent, as I've already explained. Now, the glucose transporters falls into different categories. We have 1 to 14. We have GLUT1, which are represented with the symbol GLUT, from 1 to 14 in the cell membrane. And they are tissue specific. For instance, GLUT1 is present in the erythrocyte, that is red blood cell, whereas GLUT4 is abundant in skeletal muscles and adipose tissue. Insulin increases the number and promotes the activity of GLUT4 in skeletal muscle and adipose tissue. In type 2 diabetes mellitus, insulin resistance is observed in these tissues, and this is due to the reduction in the secretion of GLUT4, which is as a result of insulin insufficiency. Now, metabolism of sugar by the colonic bacteria, because some of the carbohydrate some are moved into the small intestine. The undigested part of food, which are usually the fibers, cellulose, and all of the other sugars, are moved into the large intestine, into the colon, where the natural flora inhabiting the colon digest this starch. So digest this starch, or what we can call the fibers, cellulose, hemicellulose, and all that. Now, dietary fibers and undigested sugars enters the colon, and here the colon bacteria or colonic bacteria metabolizes these sugars to form gases, short chain fatty acids, and lactate. Now, the major short chain fatty acids formed by the colonic bacteria include acetic acid, propionic acid, and butyric acid. These acids are absorbed by the colonic mucosa cells and can provide substantial source of energy for these cells. Talking about the mucosa cells, so the metabolize. Now, the major gases formed as a result of the digestion in the colon of these dietary fibers include hydrogen gas, carbon 4 oxide or carbon dioxide as CO2, and methane gas. These gases are released through the colon, resulting in flatulence or bad or breath, as well. This is the bad breath sometimes. Incomplete product of digestion in the intestine increase the retention of water. Yes, increase the retention of water in the colon, resulting in diarrhea. When there is incomplete digestion in the intestine, so it causes the retention of water, thereby causing diarrhea. Now, we'll quickly talk about dietary fibers. Dietary fibers fall into 2 categories. We have the soluble ones and the insoluble dietary fibers. the soluble dietary fibers and insoluble dietary fibers. We have them. Then next, we'll talk about glycolysis. Glycolysis. So this is where the metabolic process begins. This is where metabolic process begins. Proper metabolism, actually. Now, glycolysis is derived from the Greek word glycose, which means sweet or sugar, while lysis means dissolution. Dissolution. And it is a universal metabolic pathway in all living cells. Glycolysis is defined as the sequence of reaction converting glucose or glycogen to pyruvate or lactate with the net production of 2 ATP molecules. Here are the salient features of glycolysis. Glycolysis takes place in all cells of the body. Know that the enzyme of this pathway are present in the cytosol, of the cell. 2. Glycolysis occur in the absence of oxygen, that is anaerobic, or it can also occur in the presence of oxygen, that is aerobic. Lactate is being the end product under anaerobic condition. Lactate is the end product under anaerobic condition in animals. Yes. While alcohol, ethanol, is the end product of glycolysis under anaerobic conditions in microorganisms and plants. In aerobic animals, that is under aerobic conditions, so to say, in aerobic condition, pyruvate is formed, which is then oxidized to CO2 and water, that's carbon dioxide and water. So carbon dioxide and water becomes the end product of glycolysis under aerobic condition. Aerobic conditions where you have abundant supply of oxygen to oxidize or to fully or completely oxidize sugars, glucose in particular. Number 3, glycolysis is a major pathway for ATP synthesis in tissues that lacks mitochondria. Glycolysis is their major source of ATP generation. Tissues like the red blood cells, erythrocyte, the cornea, the lens, cancer cells, etc. Number 4, glycolysis is very essential for brain, which is dependent on glucose for energy. The glucose in brain has to undergo glycolysis before it is oxidized to CO2 and water. So it's very important in the brain, especially the aerobic form of glycolysis. Number 5, glycolysis can be summarized by this net equation where you have glucose plus 2 ADP plus 2 inorganic phosphate and plus oxidized nicotinamide adenine dinucleotide, that's NAD, to form 2 lactate and 2 reduced NADH with 2 molecules of ATP. Then number 6, glycolysis is a central metabolic pathway with many intermediates of it, providing an entry point and as well as an exit point. Now, the reactions of glycolysis. The reactions of glycolysis are divided into 3 phases, distinct phases. We have the first phase, which is the energy investment phase, or also called the priming stage or phase. 2, the splitting phase, and then number 3, the energy generation phase or the payoff phase. We pick on the first one, the energy investment phase. Here, glucose that enters the cell from the blood circulation, from the circulatory system, from the systemic system, once the glucose are moved into the cell by facilitated transport or possibly by active transport, they are immediately phosphorylated using ATP by the enzyme hexokinase. At the carbon 6 to form glucose 6 molecules. Now, glucose 6 phosphate. Now, this is done, number 1, to prevent the glucose from leaking out of the cell, so that more glucose can be pumped into the cell. Since the glucose 6-phosphate becomes highly polar due to the negative charge from the phosphate, when this prevents it from passing through the membranes. Not only that, it is also phosphorylated to increase the energy charge of the glucose molecule, thus committing it to ATP generation, which is catabolism. Now, glucose can be phosphorylated by hexokinase or glucokinase. Hexokinase has broad specificity, meaning that it can phosphorylate all the hexose sugar, fructose, galactose, mannose. So it has broad specificity, while glucokinase, which is the isoform of hexokinase or the isozyme, is specific for glucose. There are two distinctions between there are some distinctions, I mean to say, between hexokinase and glucokinase. For hexokinase has broad substrate specificity, as I've said before, while glucokinase has limited, that is the substrate required for its activity is only glucose. Glucose is the only substrate. Glucokinase is also called hexokinase 4. Number two difference is that hexokinase has a very low Km, while glucokinase has a very high Km. Now, hexokinase having low Km means that with little amount of glucose, with just small concentration of glucose, as small as 0.1 millimolar, the activity of glucose of hexokinase are stimulated, meaning becomes active even at a very low concentration of glucose. While glucokinase has a very high Km, meaning the concentration of the glucose level in the blood has to be very high, or in the cell has to be very high before it becomes active. And the low Km of hexokinase indicate that it has a higher affinity for glucose, while the high Km of glucokinase indicate that it has a low affinity for glucose. Hexokinase is present in all tissues, but glucokinase is present in the liver and in the pancreas majorly. It's present in the liver and in the pancreas. So that's where you find them. And glucokinase will only act if the blood glucose concentration is as high as 10 millimolar. If it is as high as 10 millimolar. Now, the glucose 6-phosphate that is produced in the case of hexokinase, activity, is immediately, as I've said before, channeled into glycolysis for the production of ATP. And the standard free energy exchange for this reaction is minus 16 kilojoule per mole. And the magnitude of this change means that the reaction proceeds only in one direction, since it is highly hexagonic. Step 2 in the first phase is that glucose 6-phosphate undergoes an isomerization reaction to form fructose 6-phosphate in the presence of the enzyme phosphohexose isomerase and magnesium ion. The standard free energy exchange for this reaction is plus 2.2 kilojoule per mole, and that means it is endergonic. That means the reaction is not possible. But however, because the reactant concentration in vivo yield a value of about minus 1.4 kilojoule per mole, a value that is close to zero, indicating near equilibrium. So this will mean that slight excess of the proton can easily drive the reaction in the reverse direction by mass action. So that will form fructose 6-phosphate. fructose phosphate is isomerized. The step 3, fructose phosphate is phosphorylated using a second ATP molecule to form fructose 1,6-bisphosphate by the enzyme phosphofructokinase 1. This is an irreversible and a regulatory step in glycolysis. In fact, it is the most important committed step or the rate limiting step in glycolysis, and is the most potent activators of phosphofructokinase are or is fructose 2,6-bisphosphate, which is synthesized from fructose 6-phosphate by the enzyme known as phosphofructokinase 2. Phosphofructokinase 2. Now we move to the second phase, which is the splitting phase. Here, fructose 1,6-bisphosphate splits to form 2,3-carbon sugars, hence the name glycolysis. It is from here the name glycolysis is derived from. That is the splitting of the fructose 1,6-bisphosphate to form 2-triose sugar, glyceraldehyde phosphate and dihydroxyacetone phosphate by the enzyme aldolase, or more specifically fructose 1,6-bisphosphate aldolase. The free energy exchange for this reaction, or for the aldolase reaction, is plus 22.8 kilojoule per mole, indicating that the reaction is unfavorable under standard conditions. However, the reaction proceeds in vivo to completion because the product, that is the triose sugars, are quickly whisked away by subsequent reaction, by the next step in the reaction pathway. Hence, the rapid consumption of glyceraldehyde phosphate and dihydroxyacetone phosphate pulls the aldolase reaction in the forward direction, pulls it forward. Step 5. The enzyme phosphothioisomerase catalyzes the reversible interconversion of glyceraldehyde phosphate and dihydroxyacetone phosphate. Thus, 2 molecules of glyceraldehyde phosphate are produced from glucose. The standard free energy exchange for this reaction is slightly positive with a value of plus 7.9 kilojoule per mole, 7.9, and plus 4.4 kilojoule per mole. But the reaction proceeds in the forward direction because glyceraldehyde phosphate is quickly consumed in the next reaction. So more dihydroxyacetone phosphate is constantly being converted to glyceraldehyde phosphate. We come to the third phase now, which is the energy generation phase, or we call it the payoff phase. This phase is where ATP is produced at the substrate level. From the substrate, ATP is generated by coupling ADP with inorganic phosphate, by transferring inorganic phosphate or a phosphate group from a substrate to ADP to form ATP. Here's step 6. Glyceraldehyde phosphate dehydrogenase converts glyceraldehyde phosphate to 1,3-bisphosphoglycerate or glycerate 1,3-bisphosphate. This step is an important step as it involves the formation of reduced NAD, and a high energy compound 1,3-bisphosphoglycerate or glycerate 1,3-bisphosphate. Unlike the kinases that catalyzed reaction 1 and reaction 3, glyceraldehyde phosphate dehydrogenase does not use ATP as a phosphoryl group donor or as a phosphate group donor. Rather, it adds inorganic phosphate to the substrate. This reaction is an oxidation reduction reaction, what we can also call redox reaction, in which the aldehyde group of glyceraldehyde 3-phosphate are oxidized and the cofactor NAD plus is reduced to NADH. In effect, glyceraldehyde 3-phosphate dehydrogenase catalyzes the removal of an hydrogen atom, actually an hydride ion, hence the name dehydrogenase. Note that the reaction product NADH must eventually be reoxidized to NAD plus to ensure the continuous reaction of glycolysis so that glycolysis can continue. Otherwise, glycolysis will come to a halt if NADH is not eventually reoxidized. So what it means is that the reduced NAD that is produced is immediately transported, or if it is under aerobic condition, the reduced NAD is immediately transported to the mitochondria for reoxidation. It's immediately either via the malate shuttle or via the glycerol shuttle. So it's immediately transported to the mitochondria where it is reoxidized to generate NAD plus for glycolysis to continue. In fact, the reoxidation of NADH, which is a form of energy currency, can generate ATP. Step 7. The enzyme phosphoglycerate kinase acts on glycerate 1,3 bisphosphate, resulting in the synthesis of ATP and the formation of glycerate 3-phosphate or 3-phosphoglycerate. This step is a good example of substrate level phosphorylation, since ATP is synthesized from the substrate without the involvement of the electron transport chain. So here a phosphate group is transferred from the carbon 1 of the glycerate 1,3 bisphosphate to ADP to form ATP. Phosphoglycerate kinase reaction is reversible, a rare example among the kinase reactions. The standard free energy change for this reaction is minus 18.8 kilojoule. This strongly hexagonic reaction helps to pull the glyceraldehyde 3-phosphate dehydrogenase reaction forward, since its standard free energy change is greater than zero. This pair of reaction provide a good example of the coupling of a thermodynamically favorable and unfavorable reaction, so that both proceed with a net decrease in free energy, and which is minus 12.1 kilojoule per mole. What this explains is that the reaction catalyzed by glyceraldehyde 3-phosphate dehydrogenase is endergonic with a free energy change of plus 6.7 kilojoule, which ordinarily would be unfavorable. But as a result of the high hexagonic reaction catalyzed by phosphoglycerate kinase with a value of minus 18, it puts the reaction in the forward direction with a net free energy change of minus 12.1. So hence the reaction is feasible. Step 8 of the reaction is that the glycerate 3-phosphate or 3-phosphoglycerate is converted to 2-phosphoglycerate by the enzyme phosphoglycerate mutase. This is an isomerization reaction. The enzyme's active site contains a phosphorylated histidine residue. This phosphohistidine residue transfers its phosphoric group to 3-phosphoglycerate to generate 2,3-bisphosphoglycerate, still bound with histidine at the active site, which then gives a phosphoric group back to the enzyme leaving 2-phosphoglycerate and phosphohistidine. Step 9. The high energy compound phosphoenol pyruvate is generated from 2-phosphoglycerate by the enzyme enolase. This enzyme required magnesium ion or manganese, and it is inhibited by fluoride ion. For glucose estimation, and that is usually employed in for glucose estimation. For glucose estimation in the laboratory, fluorides are added to the blood sample to prevent glycolysis by the cells so that the blood glucose is correctly estimated. Step 10. The enzyme pyruvate kinase catalyzes the transfer of a high energy phosphate from phosphoenol pyruvate to ADP, leading to the formation of ATP. This is another substrate level phosphorylation. And this pyruvate kinase requires potassium ion and magnesium ion or manganese ion. This reaction is irreversible. The reaction actually occurs in two parts. The first part of the reaction is that ADP attacks the phosphoric group of phosphoenol pyruvate to form ATP and enol pyruvate. Removal of the phosphoenol's phosphoric group is not particularly hexagonic when written as hydrolytic reaction. Transfer of the phosphoric group to water gives the free energy value of -16 kJ per mole. This is not enough free energy to drive the synthesis of ATP from ADP and PI, that is inorganic phosphate. The reaction requires a free energy change of plus 30.5 kJ per mole. However, the second half of the pyruvate kinase reaction is highly hexagonic. This is the tautomerization. That is the shift of an hydrogen atom and a double bond of enol pyruvate to pyruvate. The free energy change for this step is -46 kJ per mole. So the standard free energy change for this reaction, or the free energy change for this reaction upon hydrolysis of phosphoenol pyruvate follows tautomerization of enol pyruvate to pyruvate is -61.9 kJ per mole, more than enough of free energy to drive the synthesis of ATP. And here is the pathway, metabolic pathway for glycolysis, the glycolytic pathway. Now we'll talk about the regulation of glycolysis. One of the first steps in regulating glycolysis is the hexokinase reaction. Hexokinase is regulated by a feedback inhibition mechanism by glucose 6-phosphate and is activated by insulin, while glucokinase is an inducible enzyme, meaning its synthesis is initiated by insulin as a result of high blood glucose level. Glucokinase is active only in the liver. And has a high Km for glucose and low affinity. Hexokinase has a low Km and high affinity for glucose. Number 2 is the phosphofructokinase 1. This is the most important rate limiting enzyme for glycolysis. ATP and citrate are the most important allosteric inhibitors, while AMP, adenosine monophosphate, act as an allosteric activators. Fructose 2,6-bisphosphate increases the activity of phosphofructokinase 1, and fructose 2,6-bisphosphate is formed from fructose 6-phosphate by the enzyme phosphofructokinase 2, PFK2. Phosphofructose 2,6-bisphosphate is hydrolyzed to fructose 6-phosphate by fructose 2,6-bisphosphatase. The activities of both PFK2 and fructose 2,6-bisphosphatase are reciprocally regulated. When glucose supply is high, PFK2 is dephosphorylated and hence activated. So fructose 2,6-bisphosphate concentration increases. This in turn activates PFK1. Pyruvate kinase is activated by insulin and inhibited by glucagon. Glucagon and glucocorticoids or cortisol inhibit glycolysis while stimulating gluconeogenesis. Thank you for listening.